So today we are starting the distal convoluted tubules. Previously we have discussed in detail the glomerular filtration, the absorption and reabsorption processes in proximal convoluted tubules, the water reabsorption in the descending limb of loop of Henle, the reabsorption of sodium chloride and potassium and calcium magnesium in the ascending loop of Henle. Now we will go for this distal convoluted tubules. This distal convoluted tubules, as I have discussed that, for example, this is nephron glomerulus, this is Bowman's capsule, proximal convoluted tubules, loop of Henle, distal convoluted tubules and collecting ducts. This distal convoluted tubules, they are divided into two parts, the early and late distal convoluted tubule. So here, for example, up to here, to this point, this portion is early distal convoluted tubules. Which type of epithelium is present in Bowen's capsule? The podocytes. Podocytes. Which type of epithelium is present proximal convoluted tubules? The brush border cuboidal epithelium. Which type of epithelium is present in loop of Henle? The simple cuboidal epithelium. Here in distal convoluted tubule, this early part is compared with this loop of Henle, the last part of loop of Henle, which is ascending limb. These both are same. Why they are same? Because this portion is impermeable to water. It's only reabsorb the sodium and chloride or electrolytes. This early distal convoluted tubules also only reabsorb the sodium and chloride, right? And parathyroid hormone dependent calcium reabsorption occur in early distal convoluted tubules. And these cells are called principal cells. And late distal convoluted tubules, these cells are called principal cells, and here are two cells which are intercalated by two principal cells. For example, this is distal convoluted tubules. In distal convoluted tubules, for example, this is principal cell and this is another principal cell. There are some special types of cells present in between these two principal cells, and they are called intercalated cell. Intercalated cell, right? This is in MCQ in your paper, that intercalated epithelium is present in, generally they are present in nephron, intercalated cells, but specifically the intercalated A and intercalated B cells are present in the distal convoluted tubules and collecting ducts, right? This is one important point. So these are, one cell is intercalated by two principal cells. This is called intercalated cell, and intercalated cells have two types. One is intercalated A cells and second is intercalated B cells. So here these cells are called principal cells. This is also principal cell. This cell is called principal cell. And for example, this cell is called intercalated A cell. This A is very important, right? And this is intercalated, or for example, this is intercalated B cell. B cell, right? And this is also principal cell, and this is also principal cell. So this intercalated cells is sandwiched by these two principal cells. They are called intercalated. But intercalated have different mechanisms for the sodium and chloride, potassium, etc. The reabsorption and secretions here, right? Because the osmolality here in blood is, for example, here it is 300 milliosmol per liter. Under this osmolality, the filtration occur in glomerulus. For example, from here, water is filtered. Electrolytes like sodium, chloride, bicarbonate, calcium and magnesium etc. filtered in Bowman's capsule. The glucose, amino acids and vitamins. Also, some of the lipids are also absorbed in small proteins also filter from glomerulus into Bowman's capsule. Then when these all things are passing from proximal convoluted tubules, this portion is called proximal convoluted tubules. We will compare all these parts now, right? So in proximal convoluted tubules, how many water is reabsorbed? Anyone? 65%. For example, from here, water reabsorption occur, which is 65%, right? Sodium reabsorption occur 65%, right? The big and main things are, right? And from here, chloride is also reabsorbed to some extent. How many? 50%, right? So these are the main things which are reabsorbing by proximal convoluted tubule. The glucose, how many glucose is reabsorbed? 100%, very good. 100% glucose is reabsorbed. Amino acids, they are reabsorbed 100%. Also lipids, 100% reabsorbed into blood, right? After this is loop of Henle. This portion is loop of Henle. In loop of Henle, this part is called descending. For example, this is descending limb, right? And this is ascending limb. In descending limb, only reabsorption of water take place because this is very highly permeable to, it is very highly permeable to water. They have which type of aquaporins? Aquaporins type 1. Aquaporin type 1. Today we will discuss the aquaporin type 2, type 3 and type 4 also. So these are aquaporin type 1. Right? From here, the reabsorption of water take place. H2. And how many water is reabsorbed here? 15%. How many? 15%. 65 and 15 is 65% plus 15%. This is 80%. It's mean that up to ascending limb of loop of Henle, 80% water is reabsorbed. And how many sodium is reabsorbed? 65%. But here in ascending limb, this portion is impermeable to water, but it has a very special type of transporter. What is the name of that transporter? Anyone? Sodium potassium 2 chloride co-transporter. This is very important. When you go in your third year, you will discuss pharmacology, and in pharmacology you will discuss the drugs for hypertension, for high blood pressure, and you will see that one of the class which is called the loop diuretics are acting on this sodium potassium 2 chloride co-transporter. It blocks this transporter, right? So these things are not only important for your papers now, these things are also important for your coming subjects like pharmacology, clinical medicines, etc., right? So this transporter here, very special transporter, and this transporter is called sodium potassium to chloride co-transporter. Right here, the reabsorption of sodium, potassium and chloride take place into blood. Reabsorption, right? And how many sodium is reabsorbed here? I think I haven't told you this. Up to 25%, 25% reabsorption of sodium occur in ascending loop of Henle, and this is impermeable to water, so 0% of water reabsorbed in ascending loop of Henle. Clear? Here, this potassium is again pumped into this ascending loop of Henle, and I have told you that this gives charges plus 8 millivolts, and these 8 millivolts push the calcium, magnesium paracellularly to interstitium and into blood. So this is the ascending loop of Henle, 25% and 65%. This is for water. And sodium reabsorption is 65% plus 25%. How many? 90%. Mean 90% of sodium up to ascending loop of Henle is reabsorbed into blood. Right? So here, which things are coming into this distal convoluted tubules? First, we have 20% of water is reaching to distal convoluted tubules, 20%, because 80% is also already reabsorbed here. And how many sodium is reaching here? 10%. Right? And chloride up to, for example, to some extent chloride is coming to this part. Also calcium is coming to this part, right? Potassium. Here, now like other cells, these early distal convoluted principal cells have also some special types of pumps here, which are called sodium potassium ATPases. Sodium potassium ATPases, and the function of these transporter is to lower down the sodium concentration inside the cell. For example, by prime reactive transport. It pumps 3 sodium out of the cell, right? And it takes 2 potassium into the cell. For example, 3 sodium out from the cell and 2 potassium into the cell. 3 sodiums out and 2 potassium in. Right? It's mean that collectively the sodium concentration is decreasing in these cells. Right? When the sodium concentration is decreasing here, this is sodium concentration, for example, it generates the concentration gradient, because the sodium concentration here is very high, it is 10%, and from here sodium is reabsorbing, so sodium concentration is decreasing in cell, it forms the concentration gradient. And here we have a very special transporter, very special, like this, but they have difference. And loop diuretics are acting on this type of channel, and thiazide diuretics are acting on this type of channel, and this channel is called, for example, this is the channel. We are sitting here, and this channel is called sodium chloride symphoter, and simply you can say sodium chloride co-transporter, but here when the sodium concentration is decreasing in this area, sodium will try to go inside the cell, and with sodium it also take the chloride ion with itself. So this channel is called sodium chloride symphoter or sodium chloride co-transporter. Sodium chloride is very high in cell. So this is active transport. This is active transport. Secondary active transport like glucose and sodium in proximal convoluted tubules. Here we have correct. We have channels which are called chloride channels, and it takes a chloride from the cell. And here we have sodium. So by this channel, sodium is going out from cell. This is the first mechanism occur in distal convoluted tubule, the reabsorption of sodium chloride. We are taking a lot of sodium chloride in our diet, salt, sodium chloride, and sodium chloride is reabsorbing from distal convoluted tubules, right? So this is the first mechanism. The second mechanism is dependent upon hormone. Second mechanism is dependent upon hormone. The parathyroid hormone function in proximal convoluted tubules is it preventing the PHO4 from H2PO4 from reabsorbing, right? Because they were inhibiting the GS receptor or the phosphate channel. But here it is calcium reabsorption. The calcium reabsorption occur in ascending limb is paracellularly. For example, we have two cell here. This is one cell and this is another cell. And ascending loop of Henle. And we know that here we have very high positive charges. So these magnesium and calcium goes or follow this paracellular transport and goes into the blood. But here the calcium reabsorption is dependent upon parathyroid hormones, and it will leave this lumen. For example, we have a very special type of receptor here. You all know, you are well aware about this receptor. This is 7-pass receptor. We have discussed this, right? From here, for example, we have thyroid gland. Thyroid gland. And with thyroid gland we have parathyroid glands, right? And from parathyroid glands, which thing is secreting? Parathyroid hormone. So this is, for example, parathyroid hormone in blood. This parathyroid hormone is activating or functioning by some signals. And what is that signal? The deficiency in body calcium level. For example, calcium is decreasing in body, right? It will signal to these parathyroid glands to produce parathyroid hormone. And these parathyroid hormone, for example, this is the parathyroid hormone. This is the parathyroid hormone. And this parathyroid hormone came and attached to this 7-pass receptor, which is GS receptor, Where this is, for example, this is ligand. This is ligand. I have told you in receptor that anything which are attaching to the receptor outside the cell domain, that is called ligand, activating or inhibiting the receptor. So this is, for example, the ligand, which is parathyroid hormone. When parathyroid hormone is attaching here to this receptor domain, it will activate the alpha, beta and gamma protein, but that have GDP, guanosine diphosphate. And when this thing is activated, this GDP will convert into GTP, guanosine triphosphate, right? And in this cell membrane we have adenyl cyclase. It will activate this adenyl cyclase, and adenyl cyclase will convert the ATP into CAMP. Which is the second messenger. And by this CAMP, pKa, phosphokinase A, will be activated. And when this kinases enzymes are activated, it will phosphorylate the transporters or proteins. So for example, we have calcium channels here. I think I have discuss this mechanism in detail, right? This is, for example, pKa. It phosphorylate these channels, and calcium present here. For example, this is the calcium which is coming from these parts, from the glomerulus, proximal convoluted tubules, loop of Henle, and this early distal convoluted tubules, right? It will reabsorbed into the cell. So for example, calcium is now present in cell, right? So it forms the concentration gradient, but remember the calcium is coming from here. It is not enough to fulfill the concentration between the blood and cell. Calcium, it is deficient now. It is signaling the parathyroid hormones, but it is deficient up to that this cell have low calcium level as compared to the blood. So here we have these transporter, and these transporters are dependent upon ATP. This is dependent upon ATP because this calcium is goes against concentration gradient. So this blood takes the calcium from this early distal convoluted tubules, and it gives sodium to the cell, right? So this is the second mechanism, which is called the reabsorption of calcium from distal convoluted tubules, which is dependent upon parathyroid hormone. If parathyroid hormone is functioning, the calcium will reabsorb from distal convoluted tubules. But if the parathyroid hormone is not functioning, the calcium will not reabsorb from this distal convoluted tubules, and we will get the hypocalcemia in our blood, right? This is another mechanism, and this early distal convoluted tubule, which is dependent upon parathyroid hormone. Now this is the late distal convoluted tubules, and this cell is called principal cell, and this principal cell is working on which hormone? Aldosterone, and the absorption and reabsorption occur in this cell is dependent upon aldosterone. Aldosterone is synthesizing in which part of the body? For example, this is kidney, and on kidney we have a very special gland which is called adrenal gland, and adrenal gland is responsible for releasing aldosterone hormone, right? This parathyroid hormone is stimulated by low calcium level, right? This aldosterone is secreting by the stimulus the low sodium level in blood or the high potassium level in blood. The low sodium level, when the sodium level is decreasing in blood, it will stimulate the adrenal gland and will release aldosterone. And if the potassium level is increasing in the blood, it will stimulate the adrenal gland and will release the aldosterone, right? Because this aldosterone, you will see now that this aldosterone is responsible for increasing the sodium level in blood and decreasing the potassium level in blood, right? So here we have nucleus. Of course, this is a cell, so in cell we have nucleus, and in this nucleus we have DNA. We have DNA, right? And in types of receptor, we have discussed the intracellular receptor. So the receptors which are present for the aldosterones are intracellular receptors. Here we have one type of receptor. For example, this is the receptor. This is the aldosterone receptor which is present in cytoplasm. This aldosterone will goes into this principal cell and will attach to this aldosterone receptor. Right? And of course this cell have this cell have the sodium potassium ATPases. All the cells have sodium potassium ATPases. And what is the role of this? It takes 3 sodium out from the cell and takes 2 potassium into the cell. Right, that makes the or generating the concentration gradient. This aldosterone, when attached to this aldosterone receptors, it goes and express some area of the DNA for protein synthesis. So, for example, these are the attacking sites of DNA, right? So, for example, from here it forms the sodium channels, sodium channel, and these sodium channels came here and attached to these cell membrane, right? For example, this area is responsible for potassium channel, and these potassium channels are placed on this lumen membrane, right? And this area is responsible for generating the high amount of sodium potassium ATPases. Sodium potassium ATPases mean that high level of sodium is decreasing and potassium is coming inside the cell. Now, this aldosterone is stimulated by low level of sodium, and when these sodium potassium ATPases are generated and placed on this membrane, it will take more sodium from here in blood, so the sodium level will be increases, right? Second thing, sodium present here how many sodium is reabsorbed? 5 to 6 5%, 5 to 6% is sodium reabsorbing in early distal convoluted tubules, right? So for example, here this is sodium plus 5, 95% sodium is reabsorbed up to distal convoluted tubule. So here, for example, into distal convoluted tubules we have only 5% of sodium, right? And how many percent of water is here? No water reabsorption taking place in early distal convoluted tubules. So 20% of water is present in late distal convoluted tubules. Now, here one another concept. What will happen to the osmolality of this area? The sodium potassium level is decreasing and the water level is increasing. It means that osmolality will be decreasing. So here we have 300 millismol per liter. For example, here we have 500 millismol per liter. Here we have 700 millismol per liter. Here we have 900 millismol per liter, and here we have 1200 millismol per liter. Here also we have 20%, but from here sodium is reabsorbing. When this filtrate is coming here in early distal convoluted tubules, the osmolality is from 100 to 200 millismol per liter. 100 to 200, and this is the reason that here the urine is very diluted, very diluted, because the sodium and other electrolytes are reabsorbing from here and water is not reabsorbing, so water level is high and sodium level is low, sodium chloride level is low. So in this area the solution is hypotonic solution, here it is hypertonic solution, and here it is isotonic solution, right? So here from this area, these are sodium channels and these are potassium channels, and here the sodium level is decreasing, and in stimulation of this decreased level, the adrenal gland release aldosterone. Aldosterone goes and binds to these aldosterone receptors, and these aldosterone receptors stimulating our expression these genes and forms the sodium potassium ATPases, also the sodium channel and potassium channel. So here we have sodium, and these sodium are reabsorbing by this sodium channels, right? And here the sodium potassium ATPases are responsible for the lowering down of sodium level in the cell, and from here these sodium will goes into the blood, and the level of sodium is increasing in blood. This is the one. The second, in response of this sodium reabsorption, potassium is going inside the cell. So these channels are responsible for taking these potassium into urine, into nephrons, right? It's mean that potassium is increasing in distal convoluted tubules, right? So these are the mechanisms which are occurring in the late distal convoluted tubules. In early distal convoluted tubules, we have reabsorption of sodium chloride, we have reabsorption of calcium, which is parathyroid hormone dependent, and in late distal convoluted tubules we have reabsorption of sodium and secretion of potassium. This secretion of potassium, because it is coming from blood into the nephron, so it is called secretion of potassium, and this is the reason why potassium level is high in urine, right? This is the other mechanism. Now we are going for this is the end of distal convoluted tubules. All these mechanisms occur in the distal convoluted tubules, the early distal convoluted tubules and late distal convoluted tubules. Now we are going for collecting ducts, and collecting ducts we have two types of cells, the intercalated cells and the principal cells.